Hi guys, so today we're talking about active transport, which is chapter 7.4 and 7.5. So active transport is using energy. Unlike passive transport that did not use energy, active transport directly uses energy to move solutes against their concentration gradient, meaning they're going from an area of low concentration to an area of high concentration. Facilitated diffusion still is passive transport because we are moving those solutes down their concentration gradient without energy. Active transport does the opposite. It uses an energy investment from the cell to move objects from an area of low concentration to an area of high concentration. This is active transport. It's moving substances against their concentration gradient. It requires an energy input from the cell, usually in the form of ATP or adenosine triphosphate. Active transport is performed by specific proteins that are embedded in the cellular membrane in order to move these molecules. This is an example of one of them, and that is the sodium-potassium pump. Active transport allows cells to maintain a concentration gradient that is different from the surroundings. An example of one of these is the sodium-potassium pump, and what it does is it uses energy to come in to bind sodium to it, to push those sodiums out of the cell, and in the same instance, so we've used that chemical energy, it allows potassium to come back in, carrying in a phosphate, so the whole process can start all over again. And if you look at the shape, you see that there's a spot for sodium and a spot for potassium. So this is one protein channel that does two things. Passive transport can either diffuse across the membrane, be facilitated through a protein, and occasionally even receptor-mediated diffusion. And then we have active transport, which always will have this ATP symbol requiring energy input from the cell. Ion pumps help maintain membrane potential. This is the voltage difference across a membrane. The voltage is created by the differences in the distribution of the positive and negative ions across of a membrane, and it is how our sodium-potassium pumps work and how our cell membranes stay functional during their job period. When you combine those two forces, you get an electrochemical gradient that drives the diffusion, passive transport, of ions across a membrane. Diffusion and active transport often work hand in hand, and here's why. First is the chemical force that creates that concentration gradient, and the second is the electrical force, which is the effect of membrane potential on an ion's movement, which can cause the diffusion to happen. This most commonly can happen during electrogenic pumps, and it's a transport protein that generates a voltage across the membrane. It's intentionally creating this type of voltage in order for something else to happen through diffusion. The sodium-potassium pump is one of the major electrogenic pumps of animal cells, and the main pump of plants, fungi, and bacteria is called a proton pump, and that's job is to move hydrogen ions. These help store energy that can be later used for cellular work, and this is something we're really going to talk about in our next chapter, because this is a proton pump, and although this is showing a cell membrane, these can be found in many membranes throughout the cell. So we use energy to pump hydrogen ions against their concentration gradient, and then through diffusion, they will slide back in, and in that process, they're actually creating this cellular energy. And that's something we're really going to talk about with cellular respiration, this idea of a proton pump moving hydrogen ions to allow diffusion to make cellular energy. We're going to skip over this. Bulk transport. Bulk transport across a plasma membrane occurs through exocytosis and endocytosis. This is the use of a vesicle or a vacuole to move large objects into and out of the cell. We know that small molecules in water can leave the cell or enter the cell through the lipid bilayer or by a transport protein through diffusion, and occasionally during active transport. But large molecules such as polysaccharides like glucose, proteins, lipids, they must move across the membrane in bulk transport vesicles. And this requires energy. Exocytosis is the process of leaving. So if you think exo, exit. Transport vesicles merge with and migrate towards the cell membrane, fuse with it, and then release their contents. This can happen because both cell membranes and vesicles have a double layer of phospholipids around the outside. This is seen most often in cells that secrete different types of things like endorphins or serotonin. They use exocytosis to export their products from the cell that then get sucked into the cell through endocytosis. Endocytosis is the opposite of exocytosis, so it's taking in of products from outside of the cell. The vesicle forms around those macromolecules and then pinches off from the cell membrane, and lysosomes come in and consume the products that were inside that vesicle. There are 3 different types of endocytosis. You have phagocytosis, which is cellular eating, so the cell taking in a solid particle. Penocytosis, which is cellular drinking or bringing in a liquid, and receptor-mediated endocytosis. In phagocytosis, our cell engulfs a particle into a vacuole, a food vacuole. Lysosomes then come in and either digest the vacuole or they digest the particle of food within that vacuole. Penocytosis, the membrane, the molecules are taken up into extracellular fluid and then gulped into tiny vesicles, and then the lysosome will come in and break them down. Receptor-mediated endocytosis is the binding of a ligand to a receptor that triggers the vesicle formation. A ligand is a molecule that binds to a specific receptor site and causes a change in shape of that site or that receptor or protein. So here we have phagocytosis, that food molecule comes in, that food vacuole pinches off from the cell membrane, lysosomes will come in and digest. Penocytosis here, that pinching of the cell membrane, bring it in, and now we have our vesicle that can be broken down by lysosomes. Or a receptor-mediated endocytosis, so the ligand binds to the receptor right here, pinches off, and then we bring those, both the receptor and the ligand, into the cell membrane. And that is chapter 7.